The TATA box is a DNA sequence found in the promoter region of genes in archaea and eukaryotes. Approximately 24% of human genes contain a TATA box within the core promoter. Considered to be the core promoter sequence, it is the binding site of either general transcription factors or histones and is involved in the process of transcription by RNA polymerase. Overview: the TATA box has the core DNA sequence 5 to TAI3 or a variant, which is usually followed by 3 or more adenine bases. It is usually located 25 to 35 base pairs upstream of the transcription start site. The sequence is believed to have remained consistent throughout much of the evolutionary process, possibly originating in an ancient eukaryotic organism. During the process of transcription, the TATA binding protein normally binds to the TATA box sequence, which unwinds the DNA and bends it through 80 degree. The AT-rich sequence of the TATA box facilitates easy unwinding, due to weaker base stacking interactions between A and T bases, as compared to between G and C. The TBP is an unusual protein in that it binds to the minor groove and binds with AI² sheet. The TATA box is usually found at the binding site of RNA polymerase II TFIID, a transcription factor, binds to the TATA box, followed by TFIIA binding to the upstream part of the TFIID protein. TFIIB then binds to the downstream part of TFIID. RNA polymerase can then recognize this multi-protein complex and bind to it, along with various other transcription factors such as TFIIF, TFIIE and TFIIH. Transcription is then initiated, and the polymerase moves along the DNA strand, leaving TFIID and TFIIA bound to the TATA box. These can then facilitate the binding of additional RNA polymerase II molecules. This cluster of RNA polymerase II and various transcription factors is known as a basal transcriptional complex or BTC. In this state, it only gives a low level of transcription. Other factors must stimulate the BTC to increase transcription levels. One such example of a BTC-stimulating region of DNA is the CAAT box. Most genes lack a TATA box and use an initiator element or downstream core promoter instead. Nevertheless, TBP is always involved and is forced to bind without sequence specificity. A genome-wide study put the fraction of TATA-dependent human promoters at 10%. An earlier study of 1,000 genes found 32% of the promoters had a TATA box. See also, Probno box, initiator element. References